Hi everyone, my name is Ericah Beason and I'm a graduate student under Astrid Schwalb at Texas State University, and I have been researching the impacts of zebra mussels on Texas Unionid mussels. Zebra mussels first invaded Texas in 2009 and are currently reproducing in 5 different basins. They're proficient in filter feeding, reproduction, and attachment, as you can see here, and they adhere to these hard surfaces after their veliger or larval stage, and these hard surfaces can include Unionid mussel shells. They can reach upwards of 100,000 individuals a square meter and deplete local phytoplankton populations. I was specifically interested at looking at zebra mussels and Unionid mussels, and these two mussels have 2 main interactions. The first being through indirect competition for things like food resources, and the second being through direct attachment, as we can see in our picture. This can lead to suffocation, decreased locomotion, and starvation in Unionid mussels. Some previous study examples. This first study found near extirpation of Unionid mussels in western Lake Erie between 1989 and 91, and that followed peak zebra mussel densities and fouling on Unionids, fouling being that direct attachment of zebra mussels to Unionid mussels. The second study saw a decline in Unionid population, and that corresponded to a significant increase in the number of attached zebra mussels to Unionid mussel shells. This last study found that glycogen concentrations tended to decline with zebra mussel attachment. When looking at ways to quantify the impact of zebra mussels on Unionid mussels, glycogen came up a good bit. It is a common metric for assessing biochemical indices, and experiments range anywhere from 7 days to 3 years. This first study found that glycogen negatively correlated with the number of encrusted zebra mussels, and we can see that in the top right. As our zebra mussels increase, our glycogen tends to decrease. They also found that glycogen was more sensitive to fouling than lipids. This next study found that as zebra mussels to Unionid mussel mass increased, the glycogen stores decreased for Lampsilus radiata. Our first objective was to examine the effect of variation and severity of fouling on body conditions, specifically glycogen storage of Unionid mussels between sites in Texas. Our prediction was that our control sites would have the highest glycogen, our high-density zebra mussel sites would have the lowest glycogen, and our low-density zebra mussel sites would have somewhere in the middle. Our second objective was to compare the impact of zebra mussel presence and fouling on glycogen storage under control conditions, so through a lab experiment. Our prediction being that our control mussels that had no zebra mussels in the tanks or attached to them would have the highest glycogen, while our zebra mussel attached tanks, this is where we had Unionid mussels and zebra mussels directly attached to their shells in a tank, and we expected them to have the lowest glycogen. We also predicted that our zebra mussels that were in the tank but not directly attached to our Unionid mussels would have some effect on the Unionids, and we predicted the Unionid mussel glycogen to be somewhere in between our control and attached tanks. For study sites, we had 5 control sites in our black circles, 2 low-density sites at our blue stars, and 2 high-density sites at our red triangles, all along the Guadalupe, Colorado, or Brazos drainages. We tried to find more zebra mussel sites, however, through countless days of scouting, most of the sites, the Unionid mussel density wasn't high enough, and we didn't want to interfere with local populations, or the Unionid mussels were dead, unfortunately. Just some images. This is control site Lakewood 1, and as you can see in the top right, we have beautiful Unionid mussels with no zebra mussels present. Here's one of our low-density sites. We averaged about 7 zebra mussels per Unionid mussel, and that was about 3 grams of wet biomass. Like our high-density zebra mussel sites where we averaged about 40 zebra mussels per Unionid at about 15 grams of wet biomass. At each site, we took a chlorophyll A sample to try and quantify food resource availability and measure that on a fluorometer. We also did a substrate composition at each site with a modified Wentworth scale, so we either had mud, gravel, sand, or cobble sites. At each site, we did a 60-minute time search to try and find the Unionids, and then followed that with transect density assessments. And then at each Unionid mussel site where zebra mussels were present, we also did zebra mussel density assessments. At each site, mussels were identified, measured, and tagged for long-term surveys, and then clipped for glycogen. All glycogen samples were taken within a 1-month time period to ensure seasonality among our sites. A 10 to 15 milligram tissue sample was taken from the muscular foot for the glycogen analysis, and all those samples were brought back on dry ice to the lab. Once back in the lab, we extracted glycogen from the tissue by homogenizing it and added an aminoglucosidase and an enzyme mix, which turned the samples a pinkish red color. That was then measured as an absorbance at 560 nanometers and compared back to glycogen standards. All of that to say, here's a nice picture. So our first two rows have this glycogen standard, and then our rows after that have duplicate wells of samples from either the field or our lab experiment. And once it turns this pinkish red color, we can read that as an absorbance and relate it back to our glycogen standards. For our lab experiment, we collected 60 Emblema plicata from Lakewood 1 and brought those back to the lab. Once back in the lab, we had 12 tanks and 5 Emblema plicata were placed into each tank. Tanks that received zebra mussels received anywhere from about 100 to 125, which is about 45 grams of wet biomass, and that was comparable to low density and high density field averages. They were then fed with commercial algae via a peristaltic pump, and that was to mimic local chlorophyll A concentrations measured at Lakewood 1. This experiment then ran for 30 days. Our field sites were similar in temperature, pH, and dissolved oxygen. However, we did have some variation in velocity and specific conductivity, but didn't have any obvious outliers. Most of our sites were finer in substrate, except for San Antonio and Guadalupe 2, and those had coarser substrate types. So again, we took chlorophyll A samples, and this was to try and quantify food resource availability. This first study found that the amount of chlorophyll in the water column decreases with increasing mussel biomass, and another study talks about algal-derived vitamins and lipids being bioaccumulated in Unionid mussels. So as we can see here, we have a good bit of variability in our chlorophyll A measurements from these field sites. Just a few things I wanted to point out. McNair 1 had our highest chlorophyll A measurement, and it was actually a low-density zebra mussel site, so I thought that was pretty neat, while we have 3 controls that have relatively low chlorophyll A measurements. The big bucks, the glycogen results. So this is for field collection, and as we can see in Belton 1 and 2, zebra mussel attachment has a significant detrimental effect to our Unionid mussels. Another thing that I wanted to point out is that McNair 1 was highest in chlorophyll A, however, it is lower in glycogen concentration than some of our other sites. Also, I wanted to point out Guadalupe 1, Guadalupe 2, and San Antonio. These sites have lower glycogen concentrations, but they were also lowest in chlorophyll A. The last thing I kind of want to mention here is that our Yegua site was second highest in chlorophyll A and our most remote site. It was in the middle of cow pasture, so I loved going to this site. But it was also interesting to see it kind of rise above the other sites in glycogen concentration. For the lab experiment, again, we can see that in our attached tanks we have depleted glycogen storage. We had about a 66% decline in energy stores between attached and control tanks when looking at mean values. We also see that we have a decline in energy stores when looking at unattached and control tanks. So we had about a 38% decline here, which was, again, very interesting to see. Putting these two together, just a few things I want to point out. So our controls in our lab experiment were relatively the same concentration in glycogen as our controls at Lakewood 1. So I think we did a really great job of trying to minimize our impact on these Unionid mussels. Again, our zebra mussel sites are in red, and again, Belton is pretty comparable to the attached mussels in our lab experiment. We also had a lot of high Unionid mortality in Belton 1, just something to note. And then lastly, our controls with the lowest chlorophyll A levels were depleted in glycogen a little bit more than our other Unionid mussel sites. We then ran a model selection analysis and found that chlorophyll and zebra mussel density was our best model. This model explained roughly 93% of the variation in our glycogen. This was the first study to compare differences in zebra mussel presence and attachment on Unionids under controlled conditions. To the best of our knowledge, we saw about a 66% reduction in glycogen between control and infested individuals. Similar responses have been seen, so 50 to 60% decline in glycogen between control and infested Unionids has been seen in the field by other studies. We can also see that attachment reduces the short-term glycogen storage, likely due to starvation and stress, which can in turn reduce long-term fitness. There wasn't much variation in glycogen between sites, which is similar to other studies, and this is presumably because they are experiencing the same stressors and same environmental conditions. Our variation in glycogen was explained by chlorophyll and zebra mussel density, and that chlorophyll was a measure of food resource, and we did see lower glycogen at sites with lower chlorophyll, presumably because they're eating less. We also know that zebra mussels can dramatically reduce chlorophyll levels. So are these zebra mussels starving our unionids? And our lab and field results would suggest that yes, they are. We saw a 38% decline in glycogen when zebra mussels were simply present in the tank with unionids, and an even more decline in glycogen when they were directly attached to the unionid shell. Some other potential stressors that could reduce glycogen and might have reduced glycogen in our study could have been higher velocity, resulting in more shear stress for the unionid, could have been runoff stressors, specifically at Guadalupe 1. It's under a bridge with a very steep slope, so maybe there's some sedimentation with high precipitation events there. We also noted high unionid mortality in Lake Belton, and there's also been notation of high zebra mussel mortality in Belton the same summer from Jason Laughlin. And so maybe temperature is at play here as well. For future studies, I would love to include different species, specifically Lampsilus terris. We know that there are differential responses to zebra mussels by unionids, but I would love to look at Lampsilus terris alongside Emblema plicata because it is a thinner-shelled individual and burrows deeper. I would also love to include riverine zebra mussel sites. So are our Texas river unionids more susceptible than our reservoir unionids? I think it would be really interesting to look at. And lastly, I would love to include other food resources, specifically bacteria, because we know that chlorophyll doesn't capture the full food availability to these mussels. I hope you've enjoyed yourself. I hope you have maybe learned a thing or two. If you have any questions or comments, please reach out to me. And I hope you enjoy the rest of your day. Thanks, guys, gals, and non-binary babes.